A number of presentations today, so we want to ensure that they run to time. Before we start, I'd like to just thank the World Organization for Animal Health for supporting this meeting. So they set up the registration of the meeting and are hosting this through Zoom for us today. So thank you very much to WOA. I would also like to just acknowledge the traditional and custodian and continuing custodians of the lands, plants, wildlife and seas. Where I'm based on Nipaluna country in Tasmania in Australia. And I'd like to acknowledge the traditional owners where you are living, learning and working today for this meeting. And we pay respects to elders past, present, and extend that respect to all First Nations people around the world that may be present. So today we're very privileged to have two presenters. We have Dr. Kelly Buckle from the Ministry of Primary Industries in New Zealand. Welcome, Kelly. Thank you. And then we also have a presentation from Dr. Paul Duff from the UK. He's from the Animal Plant Health Authority.
So my name is Kelly. I'm from the States originally. I did my vet degree in California and did a preventive veterinary medicine master's there as well. Then came to New Zealand in 2007 and did a pathology residency here in Massey University. And then in 2012, I spent the year over in Queensland, lecturing at the University of Queensland in pathology. And then I came here to MPI in 2013. So I've been here ever since, and I am a board certified veterinary pathologist, but most of my bread and butter sort of daily work is incursion investigation, as well as doing pathology input as well. So that's me. And you probably haven't met me. Those of you in the Pacific, you might or might not, because there's not that many Pacific countries on, have met these two rascals. So we've got Tom here with us today, but Andy McFadden, who's sitting to his left in that picture. These guys are undertaking a biosecurity Pacific project as part of the Pacific Partnership Initiative, which MPI and the Ministry for Foreign Affairs and Trade are funding. So you might see these guys sort of knocking about a bit in the Pacific. And they used to be on the team before they jumped ship, because they decided that biosecurity in the Pacific is a hot topic, which it is, which is, I think, what Tony and Tiggy are on board with as well. And I think we'll all agree that the biosecurity in this area really is, I mean, it's not One Health. I'm not sure exactly what the term is, but it's definitely connected between Australia and New Zealand, the Pacific. And it's really, it's hard to put a net where you keep all your birds from flying between countries, and it's hard to keep passengers from going between countries. So at the moment, this connectivity is really a hot topic generally, I think, and on everybody's mind. So moving forward, we'll go over just a couple definitions I wanted for all of us to be on the same page with. So just a note that wildlife in this context is all non-owned species. So that's including feral. And I haven't gone over pigs or other sort of non-bird species very much, but just keep that in mind as we move forward. And a disease, to my mind, and I could be corrected, but this is how I'll be using it, is any condition negatively impacting the health of animals or humans for that matter. And that can include infectious diseases, toxins. In this presentation, I'll talk about botulism, but it could be any toxin, and other causes of things being negatively impacted. So trauma, cancer, genetic disease, etc. So this topic that I've been asked to cover is wildlife outbreak investigation and how we do that and sort of the basics of it. And so I have kept it as basic as I can. Mostly my experience is the almost 10 years of investigation that I've done here through New Zealand. So I haven't done wildlife investigations in Australia, haven't done them in the States, but I will cover some of the diseases that are being seen overseas. So when I think about an outbreak, I think about the occurrence of disease cases in a population in excess of what would normally be experienced. And so outbreaks sometimes it's a question of how close are you looking, that maybe you're looking a bit closer this year, so you see a few extra cases than usual. Sometimes there is true increases. Sometimes you'll see the same number of cases, but they'll all occur together in a cluster, and so they'll look different. So it's good to know when you're talking about an outbreak, what you're talking about, and it's important to define it as much as possible so other people understand what you're talking about. So I've just sort of mangled the lovely World Health Organization one, but that's how I see it. So for instance, we're seeing in the Northern Hemisphere this year a huge extended outbreak of avian influenza that's sweeping across. All right, so how can a wildlife outbreak look? I had to think about the things that for me are triggers that make me take things more seriously. And I think these are some of the important factors. They're probably not all of them, but I think of sudden onset of disease. Sometimes they dribble in. One of the cases I'll discuss later was a little bit of a dribbling in one where a couple birds died every day for a while, but generally it's a sudden one. High numbers of dead animals or sick animals, for example, but if they're sick, you need somebody to notice that they're sick. And multiple species affected. So again, this isn't totally necessary, but it does start to increase our worry about things like avian influenza or other important disease incursions like West Nile virus, other things that we don't have here. And then just some other factors to consider, things like significant mortality over a long time, whether the disease is zoonotic, whether it spreads to domestic animals, and those sorts of things, whether it's self-perpetuating. I mean, there's a lot of factors to consider. And those are really interesting and important to think about as you go forward. And then, of course, the human health thing can't be taken, well, we shouldn't take it lightly at all. It really can't be considered too seriously. Like it's a big deal, and we definitely have cases where human health has been severely impacted by the current outbreak in the Northern Hemisphere. So just when you're considering these things, always think about human health, because it's better to address it ahead of time and to ensure you've got proper ventilation, good PPE, than to realize afterwards that you could have been exposing yourself to something really serious. Nobody wants to take responsibility or be in a responsible position after the fact for something like that. So yeah, do just have a think about the possibility that outbreaks in wildlife or any animal can potentially be transferable to humans. What are other reasons we care about disease outbreaks in wildlife? Well, they can impact the health of the wildlife generally. So the population health, conservation, and management of those wildlife. And that's really how I got into pathology, as I was interested in how wildlife were affected by disease in a conservation way. And you also, as I've mentioned, have human health impacts. So that's a huge deal. Listed a couple there, but there are way more than that. And then you have a really important potential factor, which is that wildlife might be your sentinel for a new disease entering a country. And if that disease happens to affect humans or domestic animals or other wildlife species, that might be the first time you become aware of that disease. So it is important to take it as a potentially significant indicator event. And so, for instance, in California, I've got this dead bird down here, very picturesque. In California, the first signs of West Nile virus were dying birds, and I was in vet school at that point, and you'd just see dying dead magpies along the road as you were cycling into class. But of course, the disease also progressed to humans and horses and other species of birds, and you know, it went everywhere it could possibly go. But the birds were the major way in which we saw that make an incursion. All right, and then moving on to major causes. So it's important to understand, do we have an outbreak? Is it going to kill the people or hurt the people working on it? Is it impacting other species? What sort of outbreak do we think it is? And what is the cause of it? And so the cause, of course, can be many, many things. I've got a map of Hurricane Ian that recently hit the US Gulf. So during storm events, both in Australia and New Zealand and I suppose everywhere, you've got these sort of storm-wrecked birds and wildlife that can turn up on beaches. And so you've got extra work for rehab centers and And typically, you know that that's caused by a storm event, but if you didn't know, that would be quite a dramatic thing to get to grips with. There are also toxicity events, infectious disease, of course, which is the major topic here, and other environmental factors like starvation. At the moment, we are seeing a lot of very skinny seal puppies, New Zealand fur seals, turning up on New Zealand beaches, and we're well aware of those, and we have been doing some disease surveillance for infectious disease, but we also know it's an annual cycle. And idiopathic, which means we are idiots and we don't know. So there's always those, and it's important to keep an open mind. All right, just moving on to the infectious diseases. I think I've covered most of these already, but avian influenza, of course, is the topic of the moment, and rightly so. Newcastle disease can affect multiple species, also causes rapid and severe mortality and morbidity caused by avian paraxavirus 1. Botulism, which could be also considered a toxicity, but I put it in here under diseases because it's caused by bacterial infection of the substrate. So that causes point source die-off. So birds or other animals exposed to it will develop disease really quickly, and a mass of them will die or become flaccid and have paralysis altogether. And there are other ones, and I'll go over at least one other one in a future case at the end of this talk. All right, so help, I have an outbreak, now what? And this is our world here in investigation. So first we gather data, and I think that's a take-home point people get from this. That's possibly the most important, because without data, we're nowhere. So pictures, videos, samples, if you've got them, of course, being concerned about the human health impacts of collecting bodies and making sure that you wash hands and wear gloves and things. But, most important is to really get a lot of data and get it as quickly as possible, because you obviously do want some samples and some things for to be tested if possible. So, all right, gather data, and then you can use those data. You can use those to send to people within your network, people at WOA, people like Tiggy and Tony, people who might have an idea of what is going on. So, and that happens through lots of channels. Of course, you guys will all have your own channels, but that's what we do all the time. I mean, I'll contact my mentors at Massey and say, hey, does this look like anything that you've ever seen before? What should I be considering here? And that's how you get your list. And so once you got your list, then you can start testing. So you can consider, oh, is it toxic? Is it infectious? Is it a weather event? Is there something else going on that I need to be concerned about? And then this is almost sort of a cyclic process, the way I see it, because of course, the first things you test for, or the first things that come to mind, are not always the answer, and you might have to keep looking. So just going over those three little arrows in more detail. So what to collect: photographs, videos, map location can be really helpful. A description of the outbreak if possible. So how many are normally there? How many are still healthy? How many are affected? And that will allow you to look at the percentage, so the proportion of animals that are affected. The clinical signs that they're showing: are they flopping about? Are they struggling? Or are they just dying suddenly? The location, of course, in the maps. And then what to collect is also, if possible, bodies. And those will then form the samples that you might end up having tested. And for us, we're very lucky. We've got a network of commercial laboratories around New Zealand. If we have not got exotic disease on the list, we can use those labs to help us. Otherwise, we'll get whole samples sent into the animal health laboratory. And so here's a map of a recent dead seal pup outbreak that I was involved in. So this is a little dead seal at the top. And then at the bottom, I'm not sure if you can see, but there's little red or pink X's along the foreshore there. And that is in Kaikoura, which is in the South Island. And those are little X's that were taken from GPS points that a ranger for the Department of Conservation collected. So that was quite helpful to see those seal puppies and where they were distributed along the rock shelf there. All right, and then a case definition can be useful. I won't belabor this too much, but just keep in mind, any detail you can get is quite helpful. And the more precise you can be, the better. But this will complement things like the photos and other notes you've taken. And then if you do end up taking, doing any pathology on it, that's contingent on who you've got on site, then photos of autopsies can be extremely helpful for second opinions and things like that. And save tissues in formalin and/or frozen. And of course, that is again contingent on having a pathologist or trained veterinarian who's comfortable doing it. Further establishing a case definition. So here's a picture of a botulism outbreak, which we dealt with. I forgot to get details of this case, or I forgot to get photos of this case, so I've taken this one off of the web, but this is often what we hear about, these dead birds washed up at the edge of a lake. And so in this case, the photo plus the description of hundreds of dead birds is really helpful for helping to point towards something like botulism. But if you don't have an instant differential diagnosis that comes to mind, then it can be really helpful having a case definition. It helps to clarify, to communicate with other people, helps you to figure out possible causes and ultimately to figure out what to sample. So in this case, you might actually be sampling the lake water rather than the birds. And it's not fixed in stone. So when I establish a case definition, which is, okay, what I'm dealing with, it's a mass outbreak of sudden mortality, that can change as the thing evolves. All right, so we're back to these arrows. So we've gathered data. Now I'm going to talk a little bit about using your networks. So gathering data is also about sort of a case definition. And networks, as I mentioned, are sort of variable among different professionals in different countries. There are local experts, right? So the locals, whoever lives in the village that's affected, or in the pond situation, whoever lives near the pond can tell you pretty accurately, especially if they're like an older retired person, no offense to any older retired people, but or kids who spend a lot of time there, they know what this looks like. And if this is abnormal, they will tell you. So don't discount asking local experts, people who walk their dogs along the beach and who say, oh, no, I've been walking Fluffy along the beach once a week for the last 5 years. This is the first time I've seen more than one dead seal pup at a time. This is abnormal. So that's great. WOA, of course, is a great network to make use of, and I know is very keen to be of help where possible. And then in terms of diagnostic assistance, if you do end up in one of these situations, we have got some help apparently that WOA can offer. Well, that ACDP and AHL can offer for WOA listed notifiable diseases. So there is actually the opportunity for people in Pacific countries to tap into that network and get some diagnostic assistance for WOA listed diseases. And so if there's AI on the list, then potentially there can be some diagnostic help there. But do collect all the data, because without the data, nobody will know exactly what to test for. All right, back to our arrows. So now we're thinking about possible causes. And so how do we make our list of possible causes? Well, steps 1 and 2, those 2 arrows, using your networks and collecting data will help you make your list. And if you're thinking about diseases that can cause mass outbreaks of disease in wildlife, toxicity is a big one. Infectious disease, as mentioned, environmental factors and idiopathic are pretty much the list that I start with. There are bigger lists, but this is the main one we use for wildlife. All right, and then diagnostic challenges in wildlife investigation, I think are important to touch on, because it is a little different with wildlife than it can be for domestic animals. You often don't find wildlife until they've been dead for a little while, so you sometimes have poor sample quality. So for instance, again, here's a picture I ripped off the internet, but I have seen birds like this submitted to Massey, and they are very rotten. You can see the feathers are just falling off in clumps, and they are going to be difficult to conclude what their cause of death was, although we do try as much as possible, especially with these very special species of bird. So if they're rotten, it's going to be difficult to get a diagnosis, but it's still sometimes worth taking samples and testing those samples, because some viruses and bacteria are very hardy and can actually be still useful. So yeah, you're often just left with a little bit of a difficult collection or a difficult transport to the lab. Sometimes it's difficult to get to the field in the first place. Further challenges, just to, just so that you don't drop your guard. Test validation could be difficult, especially when you are looking for antibodies to things. If you're looking just for the pathogen via PCR, then it's pretty straightforward. But if you're doing a blood test for something, it can be a bit challenging. Funding, of course, is a challenge, but we've touched on the WOA listed disease issue, so that's helpful. That there is some support available potentially. Size of the samples obtained can be difficult. If you have a tiny bird or a tiny reptile, it may be difficult without killing the animal to get any samples. And yeah, as I mentioned, there's limited available testing in some cases. So yeah, with that in mind, thought we could go into a few things that we do see in New Zealand. So just obscuring my own screen with this lovely bar. There we go. All right. And so one of the things we do see in New Zealand, as I mentioned, is botulism. We seem to have quite a lot of birds that sit on quite a lot of ponds, and when those ponds get warm and stagnant in the summer months, we end up with some botulism outbreaks. And so we do hear about those quite frequently, and we do a lot of investigations on those in the investigation space. We have poisoning events that happen. We do have a bird poison that is legal for little birds. It's very tightly regulated, but it is available to the public. And so every now and then we do hear about a bird outbreak of mortality that we then find out is related to poisoning. And then we have various other poisons that are used or that birds get into and that we end up having to investigate. So we do have various die-offs via bacterial infections, things that happen usually in the winter months, things like salmonellosis and yersiniosis. And then we do have cyclone-related seabird wreck events. So the prion wreck, it's actually called the Great Prion Wreck, I believe, of 2011. Here's one of those cute little guys. I got to help care for them. They were very cute. And so a bunch of these little tiny seabirds blew in and off the water, and I think there were hundreds or thousands of them that needed to be rehabilitated. And then, of course, the Rena oil spill in 2011 was a huge thing that affected essentially the entire global rehabilitation network. I think various people came down to help with that, and that was a huge event. So that is sort of a toxicity event, but it's in a class of its own. So those are the main ones. And then I guess die-offs in terms of starvation and things like that as well. And so I thought I'd go over a few recent cases, some mass mortality in black swans, some mortality in wax eyes, which is a little species of bird, and then some seal mortalities. So case number one, how am I going on time? Am I okay? Yeah, okay. Okay, thank you. So mass mortality of swans, here's some lovely healthy swans. Now, swans are not, I think, particularly worried about native species, but we do worry about mass mortalities. And so this is where we come to swans from, is worrying that they might have been affected by an exotic disease or by something contagious that might affect other species. So we had a local Fish and Game official that phoned MPI. We have a 0800 hotline that people can do this through. So he phoned to report that there were dozens of black swans on a sewage treatment pond. Now, although it is a sewage treatment pond, these birds apparently usually are quite healthy and the pond itself is monitored. So this was unusual. Only the one species was affected. So no eels, no other birds, just the swans, which was a bit unusual and strange. And they all occurred over 1 to 2 weeks in the late autumn. So that's kind of fitting with the botulism event, but the single species was not fitting as well. So he was a bit concerned, and because it was so many swans, our radar was a bit heightened as well. All right, so we gathered some data. We went and got, the lovely Fish and Game guy went and got some bodies. He got the freshest bodies he could. He gave us some timing and season information, which I've shared with you. And he was one of the local people that I suggested you talk to. And he also knew the people who were in the sewage treatment pond business, and they could tell us that this did not happen normally. And so, yeah, we spoke to our scientists here at the Animal Health Laboratory, and we talked amongst ourselves, and we determined that it was most likely botulism, but we weren't 100% sure. We decided the weather probably didn't play a factor except in potentially promoting botulism. So we were left with toxic and infectious. We do, as I mentioned, like to, we do like to, I'm not sure what's going on here. Oh, there we go. Okay, we do like to exclude exotic disease. And so, so we did that in this case. And we, yeah, so the local Fish and Game, I think I went over this in the last slide, but he said it definitely was not normal for this time of year, and he helped collect the bodies. And we tested for botulism, avian influenza, and Newcastle disease virus. We decided those were the most likely exotic ones if there were any exotics. Botulism, of course, is not exotic. And then we did histology on the swans as well. The slides are taking a little while to progress. Yeah. There we go. All right, and so we did the testing and all of the exotic disease assays were negative, which of course is great. Unfortunately, the botulism was also negative. Now, this could have been a sample dependent thing, and we weren't able to go back and get a second set of samples. So this is the one question mark of this. We still consider it a probable botulism case. Histology, though, showed high levels of parasites, which was interesting, and we put that in our database and actually got those parasites ID'd so that we know what's going on out there, so that we can see if anything changes the next time we do get histology from swans. And so we were able to exclude exotic disease, but we weren't really able to come to a final conclusion, although botulism is considered most likely. All right. And then our second one here is we did actually get an answer, which is very satisfying. This is an adorable wax eye. This is another photo that I've taken from the DOC website. A member of the public in Auckland phoned the MPI Exotic Pest and Disease Hotline. And he was very concerned because around his house, there used to be lots of little flocks of wax eyes. And he started finding 2 to 3 dead wax eyes daily for a few weeks, which is kind of a lot of wax eyes over time. So he became concerned. Originally, he thought, oh, it's just a winter mortality event. It's just because it's been very cold, and it had been very cold and rainy. But then the winter weather sort of warmed up and the sun came out and they kept dying. So that was when he decided that we should be called. So fortunately, he'd kept 2 very fresh bodies in the freezer, double bagged, which is very helpful, since you don't really want fresh dead birds mixing with your other things. And he was able to get them to us for postmortem. And the samples were, so exotic disease was very low on the list, and endemic disease was very high on the list because of the long, slow nature of this. So we did get samples taken for exotic diseases, avian influenza, West Nile virus, and Newcastle disease, but we were sort of assuming it was more for surveillance than anything. We did, however, we were lucky to get some great lesions. As a pathologist, I appreciate these, and although nothing was seen abnormally on autopsy, the lesions on histology were spectacular, including this guy, which is a huge sort of quote unquote abscess, because birds don't do abscesses really, but in the brain. So you can see this guy probably had a bit of a headache. And if you look a bit closer, these bacterial colonies, which are sort of purple and finely granular, look very much like Yersinia. And this is kind of what Yersinia does as well. So the pathologist who was doing this, who wasn't me, did a gram stain and it was gram negative, which is consistent with Yersinia. And so that was very nice. All samples were negative for exotic diseases, but there was this severe bacterial infection. And that was in both birds. So we can assume that possibly most, if not all of those mortalities was associated with the Yersinia infection. And this did eventually go away. And so we closed it. So that was a nice one. And then just one note about seal mortality events. This is the other wildlife species that we do see mortality in. And we do sometimes see that it's due to trauma, either being washed up on the beach or occasionally by human hands. Unfortunately, we do see a lot of starvation just as a natural event during weaning. So it is a cyclical annual event. because those little puppies get out there and the waves are rough and they have a hard time foraging. And so unfortunately, they don't make it through the winter. And DOC mostly deals with the ones and the twos of those. If there are 3 or more that are seen, they do come to us and we'll do our investigation and exclude avian influenza and morbilliviruses as possible. So that's what we do for those ones. And so this brings us close to the end of this talk. Thank you for your attention. The take-home messages are, from my perspective, are that it is really useful to investigate wildlife outbreaks. It's not a waste of time. It's not a weirdo thing. It's actually a core part of business while looking for exotic disease and looking for animal health. Collect as much information as possible. Phone a friend. Which is what we do all the time. You know, pick up the phone and just go, hey, what do you think about this? Do you have any other ideas? Collect the freshest samples possible, not the rotten ones if you can help it. And then do consider involving WOA and the local country experts for help, even if it's just advice in the early stages. I thought I'd throw in a few resources for avian influenza just because it is such a hot topic. And there are some really excellent websites out there. Thanks to Tiggy and Tony for throwing a couple of these at me. These all sort of link together, so I didn't want to put too many because eventually you end up at the same sites. But these are quite nice ones for having quite a few resources for human health and managing it from an avian perspective. And then Tiggy and Tony have tried to define what will happen with samples. And does one of you would one of you like to speak to this? Should we just leave it?
Kelly.
Oh,
yeah, it's fabulous, Kelly. No, that was a fabulous, absolutely fabulous presentation. And this just really sums up the diagnostic support that might be available for those countries in the Pacific that might require some additional assistance, for example, from Australia and New Zealand. And certainly this, you know, so it's around phoning a friend and how that might work specifically for WOA listed diseases and through, you know, through discussions through the government authority. You know, we're not going to, you know, member of the public won't phone up somebody in Australia to help out. And as Kelly has clearly outlined in her presentation, you know, any investigations that are supported by Australia and New Zealand, it's really helpful to have all that additional information that's available as part of the disease investigation, because it really limits being able to investigate it appropriately if you don't have all of that information. So that sort of, you know, is criteria that's sort of required to sort of initiate a support system for diagnostics. So if there are not any other questions at this stage, I'll maybe hopefully share my screen and the next presentation, you know, correctly. But we have Paul Duff, who is from the Animal Plant Health Agency in the UK. He was the WOA focal point for wildlife for many years, and he's currently doing a PhD. He will introduce himself again. So I will share my screen and hopefully please keep me straight if I don't get this right.
Hello, my name is Paul Duff. I work for the Animal and Plant Health Agency. And together with several partners, we provide national wildlife disease surveillance in the Diseases of Wildlife scheme. My co-authors here are Craig Ross and Rowena Hansen, who are both virologists, and Bart Donato, who is a wild bird ecologist. This work occurred in the winter of 21-22, last winter, and it's a study of mortality due to H5N1 high-path avian influenza infection in wild geese in a estuary in England. Two aspects were apparent in this study. The first was that this was an unprecedented mortality by our standards nationally in England and Wales, and it was due to an infectious agent, which was slightly surprising. The second aspect was the difficulty when investigating notifiable disease when at the same time you're attempting to take a One Health approach, in particular to assess losses due to biodiversity. Why is high-path avian influenza so important? Firstly, at the top right, you can see the losses due to domestic poultry. So this is the largest outbreak ever recorded, and you'll see, comparing to other years, that this is significantly greater than previous outbreaks. Many of these domestic poultry flocks are huge, and the overall cost is in terms of millions of pounds. I tried to find some figures to assess this, but they weren't available at the time. But it is costing a lot of money. As far as wild birds are concerned, we're looking at one species here in particular, the barnacle goose. There were 57 confirmed deaths due to H5N1. 45 of those came from this estuary, which is called the Solway estuary. In our field trips, we counted 750 dead barnacle geese, and we recovered 40 rings. The flock size is 40,000 birds, and professional zoologists reckon that by the end of the winter, there were 10,000 birds missing. In addition to this, we're aware that RNA virus is always new and emerging. It's always producing a potential for zoonotic disease, and in fact there was one human case in England, asymptomatic. There's the threat to farm animals, in particular for poultry, and in this disease wildlife remains a constant reservoir, unlike, for example, COVID, where bats may have introduced the virus to humans, but it's human-to-human propagation that is important. Migrating birds spread the virus almost globally. And finally, there's a tendency that this virus is becoming more important each year. The barnacle goose is IUCN least concern. There are several populations as indicated on the map, but the one we are looking at is within the red ring. And here the birds breed in the high Arctic, in Spitsbergen and Svalbard, and they migrate to this one Solway estuary. The flock started in the end of the Second World War, 1945, and it has increased steadily in that time to the present population of about 40,000 birds. So the Solway estuary is between Scotland and England. The dark green areas are the salt marshes where the barnacle geese feed, and they may visit these each day. The important thing is that the flock, the wintering flock, does not tend to move out of this estuary. Other migrating geese and water birds fly in and fly out, but the barnacle geese are resident here from about November through to May. The middle map shows you the rough extent of the estuary, and the map on the left shows the density of HPAIV infections during last winter, with densities increasing along the western and eastern shorelines. So what did we do? Well, with local ecologists who asked me to come and help them with their investigations, we undertook visits to one marsh in PPE, personal protective equipment, every month or every 5 weeks. We saw strandline mortality of barnacle geese. We saw that the birds fed and roosted and flew in dense flocks. And we also saw that birds disappeared quickly, either through predators or through the tides or through the mud. How do we process these birds? Well, this has been published last year, and these are the detailed steps. But in summary, if the public or anyone finds sick birds together or dead birds, they phone a single helpline for the country. The dead birds are collected by a special agency, and they are brought to the nearest regional laboratory, which in this case was about 30 miles away. At the laboratory, in containment level 3 cabinets, the birds are swabbed. The swabs go to the Central Veterinary Laboratory for HPAIV PCR tests. If those tests are negative, we are then free to get the frozen carcasses and do diagnostic post-mortems. And finally, we send reports out to the people who made the initial phone call about the dead birds. There have been reports of 7 species of wild carnivore that have been affected by H5N1 last winter, and they are listed there to the left. On the photographs, we had a fox submitted from one of these marshes. I examined this animal. I found that it was heavy and in very good condition and with good deposits of fat. Its stomach was full of dark bird feathers and fat, presumably of these geese. The photograph to the bottom right shows one of the geese which I examined, and the pathology in both barnacle geese and pink-footed geese, which this is, was the same as that's described in literature, including in some publications by the APHA. So it varied from very subtle pathology to splenic necrosis, pancreatic necrosis, lung edema, etc. Now the histo pictures to the top right are from an outbreak of HPAIV in the previous winter in a wildlife rescue center, which affected a fox and 2 species of seals. They show heavy labeling of all in the CNS of all those mammals, while the labeling, ICC labeling for virus in respiratory tissues was relatively low, and in some of those cases respiratory swabs were negative for virus. We use the word unprecedented in the title, and in 24 years of national wildlife disease surveillance, this incident by far produced the greatest mortality. Here we're describing incidents as a single location with 1 or 2 causes and a mortality greater than 100 deaths within defined time periods. So the next largest mortality was in 2013, and that was 4,000 birds dying in the English Channel from chemical poisoning. On our visits, we counted the carcasses and we sprayed the feathers of the carcasses a different color. And we did these between November and May, and we could see feathers of the original geese from the original visit on the last visit. So this spraying did work and prevented double counting. And importantly, it showed that within this flock of 40,000 geese, the clinical course, assuming that most of the birds were going to be exposed to virus, was 3 months. Escalation and mitigation. These were ad hoc reports. They weren't officially requested, but I did think that it was important to get this information to the policy personnel. They were dealing with the big challenges about preventing poultry flocks being infected. So we produced 5-page reports with photographs and with as much data as we could. We didn't offer anything in the way of conclusions or advice. It was simply to get information to the people who were trying to control the disease centrally. So this can be quite dangerous because the news is not good. You're saying that other species are infected, wild species are infected, and you're telling this to people who are really overworked and overstretched with the challenges in the poultry industry. Issues we were aware of were that the public, taking matters into their own hand but against advice, collected carcasses on a few instances and put them in open plastic bags. We put about 26 carcasses in these bags and left them on the beach. So this was not what we wanted, totally contraindicated. On the other hand, the local authorities produced very good information posters. They placed these at conspicuous areas and car parks along the beach. And basically what they are saying is, don't approach these birds, don't touch them, don't let your dogs go near them, wash your hands after each walk, and if you see sick or dead birds, phone the helpline phone number. Start the process going. So the outbreak is still going as we talk here in September, October 2022. There are still outbreaks in wild birds and there are outbreaks in poultry. New protection zones have occurred this week. So these are provisional observations. The first is that we are really poorly sited and poorly prepared for HPAI-V risks to wild birds. Second point is that the laboratory confirmed cases give little indication of the total mortality in some of these species. Wild bird migration patterns are very variable in time and geography, and these barnacle geese stayed at the winter site into May, and some of them were still around in June, presumably too sick to migrate. Can affected wild birds and wild bird flocks be managed? Or should we wait for a flock immunity to occur? Is that the best conservation approach? These were the questions that were being posed. Some provisional conclusions. We see this new challenge, which we've never really seen before, in addressing notifiable disease in wildlife, where it's causing significant mortality in the wild species. There aren't that many notifiable diseases in wild species in England, and this sort of issue of mass mortality is very unusual. So in these circumstances, is the One Health approach practical, where nationally, the important thing is to prevent domestic poultry being infected? Our experiences suggest that you need to monitor early. They indicate that you must work with ecologists who have specialism in those species. I feel that it's important to try and get the ecological data back to government and to policy. And we also found that it was useful to publish open access to provide awareness for the future. It was incredible how quickly these carcasses and feathers disappeared. There's no remains of those birds now, but these carcasses disappeared quickly, so you have to work quickly to monitor what the disease is doing. Finally, I'd just like to express my thanks to the APHA and to DEFRA, the Ministry of Agriculture in England and Wales. I certainly would like to express my thanks to ecologists who work together with us. And I'd like to take a moment just to acknowledge the help of Paul Holmes and Alex Barlow, who've helped me for 24 years in the Diseases of Wildlife scheme. Thank you for your attention,
And chat to you all next time. So thanks, everyone. Have a good rest of your week, and chat to you soon.
Thank you so much.
Thanks, Tiggy. Thanks,
Bye.
Tony. Thanks, Kelly.